everyone, welcome back to my classes. Today we will discuss about the nature and source of drugs. So what is a drug? So this term drug is been derived from a French word which is called as drogue, which indicates a dry herb. So drug is nothing but any chemical entity which is present in a medicine which is used for the diagnosis, prevention, treatment or cure of a disease. So WHO has been given a standard definition for the drug. Like drug is any substance or product that is used or is intended to be used to modify or explore physiological systems or pathological states for the benefit of the recipient. So whoever is taking this particular medicine, either it is going to change the pathological condition, nothing but illness is pathological condition, or if any system of our body is not working properly, physiological system, then it will explore it or it will alter the function. That is called as drug. So every person in their lifetime, they are exposed to several drugs. From where these drugs are obtained, from where they are being prepared, that is the nature of the drugs. So the drugs can be available either naturally, which are available from the nature, synthetic, which have been prepared synthetically in the laboratories, or semi-synthetic. Semi-synthetic is nothing but we are taking a natural product and we are modifying it. So we are terming it as semi-synthetic. So the nature of the drugs are natural, synthetic or semi-synthetic. So we have seen that drug can be available naturally, synthetical or semi-synthetic. So what is the source of this natural products? From where what is the source? So natural products or the natural drugs can be obtained from the plant source, animal source, mineral source and from the microorganisms which are available naturally. From them we are going to prepare the drugs. So that is a natural source. Let's start with the plant source. So from the plant, roots, leaves, seeds, from any part of a plant we can extract a drug. So the most commonly available active ingredients are glycosides, alkaloids and castor oil. So glycosides, they are obtained from the cardiac glycoside from the digoxin or foxglove plant. From the alkaloids are prepared from the morphine from the poppy capsules or atropine from the belladonna seeds, or quinine which is obtained from the cinchona tree, and the castor oil which can be obtained from the castor seeds. So in the images I have mentioned about the vinca plant and the morphine and the castor seeds. So from the animal source, we can take hormones like heparin, insulin and thyroxine from the pig or the ox, cod liver oil from the fish cod liver. Next source is microorganisms. The most commonly seen antibiotics which are prepared from the microorganisms. Few of the examples are penicillin from Penicillin notatum, streptomycin from Streptomyces griseus and bacteriacin from Bacillus. All the three images are being mentioned here. Some of the naturally occurring minerals include magnesium, calcium, aluminium, sodium, potassium, iron salts, inorganic metals like lithium, iodine, and the radioactive elements like iodine. Moving on to the second type, semi-synthetic source. So whatever the natural drugs they are available, we will take them and modify it synthetically. So we call it as semi-synthetic because we are taking a natural one and modifying it synthetically. So we term it as semi-synthetic. And the examples are genetical engineering and the human source. Genetical engineering. This genetical engineering is nothing but we will refer to the direct manipulation of the DNA. This genetical engineering is also called as genetic modification or genetic manipulation. Nothing but we are breaking the DNA strand. Gene splicing. We are replacing that breaked DNA with a new gene in that. So modifications in the genes is nothing but genetical engineering. To the human source, from the pregnant females we can obtain the HCG, whereas the postmenopausal females urine we can take the minotropin. From the human we can take a regular insulin. From the human kidney cells we can separate urokinase, and we can have the recombinant human growth hormone. RHGH is nothing but recombinant human growth hormone. All these are the human sources. If the drugs are prepared or synthesized in the laboratories, we term them as synthetic drugs. So once we know the active ingredient or the plant drug, the structure of the active ingredient will be determined and we can start preparing them in our laboratories, which will be synthetic drugs. Examples like sulfonamides, salicylates, barbiturates, all these are the synthetic drugs. So the synthetic drugs, when you are preparing, the quality can be controlled and the process of preparing is easy and the cost will be less, and they are safe to use, they are more potent, and we can do large scale production. So the advanced therapy can be given by using the hybridoma technology and stem cell therapy. Hybridoma technology is also termed as monoclonal antibodies, nothing but here the antibodies are being produced, similar antibodies. Monoclonal means, mono means single, so same type of antibodies have been cloned. So it is a method of producing larger number of identical antibodies. Whereas the stem cell therapy, the stem cells, they have a capacity to build every tissue in our human body. So the treatment with the stem cells is also called as regenerative medicine, which will promote for the repair response of the diseased, dysfunctional or injured tissue by using the stem cells.